finish their drinks and sandwiches. The timer didn't start yet, right? All right. Thanks, everyone. So getting there. Yeah, I was. Yeah. Yeah. We should do stuff together. Yeah. All right. Well, things quiet down. I will just start to present what I would like to share with all of you today. We are here working on development, biosimulation testing, and manufacturing medical devices. Oops, sorry, that makes a funny noise as well. And I actually have two company names there at the top of the slides, and I would like to take 10 seconds to explain why that is. Resolution Medical is a company founded in Minneapolis a while ago. They are focusing mostly on device development, delivery system development from a very early prototype stage all the way through the full manufacturing. And they work not with their own brands, but they work as a contracted party to people that have great ideas but don't have the facilities to build these devices themselves. And LifeTec Group is a company that I co-founded in Eindhoven almost already 20 years ago. Our focus has been mostly on how to simulate ex vivo what will happen when you have a device that is in a stage where you might want to try it out. So simulate what will happen if you go in vivo or if you go in animal or in human later on, but already at a very, very much earlier stage in that process. And we were actually doing quite a lot of work for startup companies in the US, so we're traveling back and forth all the time. So at some point we were looking for our own footprint in the US to have our own lab. And that's in that phase we met with Resolution. And since our services were so collaborative, I would say, we were offering our services to the same customer base. We decided to join forces. And in the end, 2 years ago, they acquired our company. So now we have lab facilities there as well. So I'd like to spend a bit of background on what Resolution does and how we fit in later on. As I mentioned, device development, mostly cardiac and vascular. So the usual suspects in clinical fields are listed there. Electrophysiology and neuromodulation is actually not that far apart. From a clinical perspective, yes, but if you look at it from a device technology approach, it's leads, it's stimulating tissue and measuring back what the tissue is doing. So devices in that space are actually quite similar. And if you look at all those fields and how we interact between the two companies, then first off, you have a great idea. You want to build a prototype. Our team in the US does concept engineering, like very early prototypes. If you want to make a heart valve out of a paper clip and a chewing gum, they can do that for you. And in that early stage, there is no formal stuff, no documentation, no quality, just really MacGyvering. And that's where biosimulation actually fits in. If you have a concept prototype really early on and you want to try how that works, we think giving you a very realistic environment in which to do that and focus on feasibility of the delivery, deployment, and stuff like that, that comes very nicely along in that early stage. So we can also quickly iterate. You develop something on your bench, you run to the lab, you try it out, you go back. You can't hear? I'm too tall? All right. Sorry. Yeah. We'll do the beer part later, okay? So I was trying to explain that very early prototyping and biosim feasibility testing fits really well together, and especially can speed things up greatly, because by the time that you have a prototype and you want to go to an animal lab, it's 2 months down the road that they're available and you won't learn as much as you could in a lab setting. But once you've passed that phase, then the logical next step will be formal product development, and that's where documentation kicks in, quality management kicks in, regulatory kicks in. And in that phase, you will build, let's say, about 100 devices, and you go do your first in vivo stuff. The next phase in there would be, if you can build those prototypes for your earliest trial phase, then you need to think of how am I going to build 100,000 of them instead of 100? And that's where transfer to manufacturing, large volume manufacturing process kicks in. We have a specialized team to cover that. And at the end of the road, there's always, obviously, the large-scale manufacturing business. Now, we have some capabilities here from early design, anatomical models. We have metals forming groups. Nitinol team is really good. We have laser welding, cutting, all those kinds of things that you need to actually build prototype devices and delivery systems. Obviously, the testing equipment is there as well. We have the regulatory support covered. We are ISO certified, FDA registered. We have a quality management system. Basically, everything you need in one house. But I'd like to focus what we do in biosimulation, and that's kind of where our company started with. So the purpose here is to mimic as close as possible what you might find in vivo in an early stage to bridge the gap between bench animals and patients in the end. Now, this graph kind of explains where I'm going to get at. If you have an idea for a prototype or for a device, then if you look at this whole pathway, you have lots of different steps, and every time you advance to the next step, it becomes a bit more time consuming and a bit more costly to finish that phase. And if you look at the most important bit in that is lab testing, animal trials, and clinical studies. And with a frozen design, you have to pass all three of these stages. But as you can see in the icons there below, running a test in a lab on a machine is, for the same device, very different from machine to animal. Not per se for the device, but the material properties and everything around the device changes quite a lot. So that machine, that animal has completely different. I'm getting lost here a bit. Sorry about that. It's so quiet all of a sudden. Well, it's tissues, it's warm, it's biological tissue, it's a different approach, the different imaging, everything is different. And also from animal to human, you might think that's not such a big step, but you go from young and healthy animal tissue to sick and elderly diseased patients with pathologies. And the team that is involved in the studies is also all of a sudden completely new. It's physicians instead of engineers who were at the table in the animal lab. So you have to run through all these phases with one device design. And even though you have three completely different test environments, we still hope that the end results will be at least comparable, right? So what you want to prevent from happening is that you end up in vivo and find out that the results that you were hoping for is not what you're getting. And then you have to start iterating in those costly, time-consuming phases. So what we think we can offer with biosimulation is to mimic what happens in vivo with real tissues in a realistic environment, but already in a lab phase, so that you can be de-risking the next stages and be sure that you can do what you expect that you can do once you go in vivo. Now, for that, we've developed quite an array of, let's say, biosimulation technologies. Most of them are including tissues, human or animal. Some of them are even perfused, where we're trying to keep the organs functional and alive. But let's focus, for the sake of time, on our cardiac applications. I'd like to present two setups here. One of them is the cardiac biosimulator, which is based on cadaver tissue. So these are not alive, but they are anatomically correct, of course, since they're the real deal here. And these are already great as a first step to look at delivery approach, navigation of devices in the heart, deployment, anchoring, imaging, and also the procedural workflow of the whole interventional procedure, basically, right? And we're looking at real anatomy and real tissue properties. Now, the next model is very much the same in terms of what is around the heart, but the major difference there is that we treat the heart as if they were donor organs, and we actually revive the hearts, so you're looking at living hearts. there. It's way more complicated than the cadaver approach, which is why we don't offer that as a first step. But if your therapy or your device, once it's inside the heart, has some kind of interaction with the living properties of the heart, for example, the contractility of the muscle, creating forces on your device, or you're trying to interact with the electrophysiological system there, then the living heart has that, where the dead heart does not. So just to explain what we're talking about, you see here in this little clip, a heart, a real heart, a pig's heart in this case, on a bed. We actuate the ventricle of the heart, so we have a big cannula there in the apex of the left ventricle in this case, where we apply a stroke volume. While we're displacing fluid through the heart, the valves start to work, which you can see on the video there, which is maybe more insightful than looking at this from echo or fluoroscopy. Already you can see that. The circulatory model around it allows us to tune blood pressures on the venous side, on the arterial side, but also inside the heart. We have hemodynamic measurements, so we can measure pressure and flow signals throughout the system. And you can also assess if we create a diseased, let's say a failing heart valve, you try to repair that with your prototype device. You can see how that hemodynamic response is to that intervention. Also, we don't have a body around it, which gives us great freedom in directing the vasculature, so the approach pathway into the heart. We can make that look like animal. We can make it look like human, which is significantly different, I can tell you. As I mentioned, compatible with engineering measurements. For example, the camera systems that we have in there are not something you can do in vivo, but they're just so much more intuitive in trying to assess what you're doing. Of course, we can also apply the clinical measurement techniques that you would use in a hospital. It's quick to exchange hearts. It's within 5 to 15 minutes. You have a new heart on the table. You can repeat your procedure, so you can do multiple in a day. As you can see in the pictures here, the system is very much configurable to whatever you need. The top right picture is us at our own booth in EuroPCR, so it's a small footprint platform there focused on ultrasound visualization. In this case, we were doing some clip procedures. But at the bottom right, you will see the completely different layout of the exact same system mounted in a C-arm on a bed, like we could possibly do here in the imaging lab next door. Left heart models, right heart models, any approach that you can imagine, we can add vascular models. We can mimic pathologies. We can dilate annulus to create valve leakage or anything. Basically, we can try and recreate that setting as close as we can. And since we're mostly working with animal tissues from slaughterhouse animals, we don't have any ethics to deal with, so that can prevent us getting delays and getting protocols approved. Examples here. This is endoscopic view from both the ventricular and the atrial side of the tricuspid valve. We're looking at a tri-clip procedure. I'm going to walk here to point. So this is that clip. We created a dilated annulus of the tricuspid valve, so you will see that the valve leaflets are not exactly closing anymore in the middle, and that's something we try to treat with this procedure. So that clip is kind of like a washing pin, tries to grab the two leaflets while they're moving in the heart. We have Philips's ICE visualization cap, so that's ultrasound on a catheter tip. That's already out in the US. It's not in Europe, approved yet in Europe, I should say. You can see the ultrasound imaging there at the bottom right, especially for tricuspid interventions, TEE, so transesophageal echo, is very difficult to see that deep into the heart, and that's why ICE is the next thing on the market. But just to show what it looks like, you can imagine getting entangled in the chordae, all those little cords that hold the valve leaflets in place while they're closing. If you get entangled, it's going to be a mess. You can see that here. You can see that you're getting entangled. You can try to bail out, but you can also see on the ultrasound, you don't see the cords. You're lucky to see the valve. So from a clinical teaching perspective, that is also something that it can be very important. Know what you're doing, know what you feel while you're looking at your therapy inside the heart. The next example is looking again from the ventricular side, but in the atrium, we're looking at the transeptal puncture that we did as an access pathway. In this case, we are using 3D TEE ultrasound. We have also, the video is not really moving as nicely as it was before, but we have fluoroscopy imaging there as well to show how device location is inside the heart. And you can see here the 3D TEE allows you to see the location of the device within the annulus of the valve, but you don't see the valve structures that well. You don't see the cords that well. So especially if you're initially figuring out how to navigate and where to deliver or deploy, this is a great tool, I think. Examples of use, I've already mentioned, early device, prototype stage, feasibility of the procedure, but also clinical training. And we've, well, worked with so many device types already. I'm not going to run through them one by one, but if you have more questions, you know where to find me at the back there. This one is the other model, the physio heart model. We revived the heart so you can see if it's running. Yeah, okay. We reperfuse the heart on whole blood after having treated the heart as if it were a donor organ, and we revive it. You see this is a real living, contracting heart just outside of a body, which gives you a lot of freedom to play and to interact with that heart. As I said, reperfusion on blood, contractile myocardium, electrophysiology is functioning. And this is, as I explained in the introduction, really useful if you want to interact with the living components. For example, if you want to place a VAD, an assist device, you can make the heart fail, and then you can put the pump inside and see how the heart responds to that treatment. That is, for example, not something that you can do with a cadaveric model because it's dead. It won't respond anymore. Okay. Well, lots of applications. In light of time, I have that video running at the table there as well if you're interested. So, but the bottom line here is we are here to help you. For biosimulation, well, you could ask what's in it for you. Mostly learn what goes wrong in an early stage, right? Learn it early on, iterate quickly with design improvements, and make sure that you de-risk the expensive stages of your pathway to the market. Know that your therapy will be functional in animal before you go there, and use less animals. For example, if you have 3 device designs that you are considering, you can already select the one that you want to go in vivo with, and thereby save quite some animals. Well, as mentioned before, training is something that is specifically valuable for clinical teams. In terms of device development and manufacturing, we're not the only one out there. We don't do everything, but we're good in complicated devices, steerable devices, especially delivery systems, and implants, even active implants like pacers and neuromodulation devices. We don't do everything, as I said, but we are focused. Because we're focused, we can be quick. We're happy to support, even on sub-assemblies. If your team should focus on the complicated parts of your device and you need someone who help you out with a sub-assembly or delivery system, we're happy to do just that. We actually try to be a part of your team until you're where you need to be in the hospital. And that's what I had to present here today. Thank you. If there's any questions, I'm at the back there.